Bismillahirrahmanirrahim. Assalamu alaikum dear students, welcome to your channel The Anatomy Canvas. Today we are going to discuss the histology of the salivary glands. So learning outcome for today's class are that to identify the variety of the salivary glands according to the nature of the acinus, to study the stroma and the parenchyma of the glands and describe the duct system and differentiate between the ducts. So structure of the salivary gland, you can see in this diagram that the salivary gland consists of the end bulbs of the secretory units. These secretory units may be serous end bulbs or maybe mucous end bulbs, and their secretions drain via duct, smaller duct into a larger size ducts. So how we classify the salivary glands? So salivary glands can be classified on the two bases. First, they can be major salivary glands and the minor salivary glands. And on the basis of the secretory products, they can be classified as the mucous salivary glands, the serous salivary glands, and the mixed salivary glands. So major salivary glands are three pairs: the parotid, the submandibular, and the sublingual glands. Saliva secretion is nearly 600 to 1500 ml per day, which conveys by the ducts to the oral cavity. And percentage of the saliva is that parotid contribute nearly 20%, submandibular contribute 70% to it, and sublingual contributes 5% to the saliva, and the minor salivary glands contribute nearly 5%. So minor salivary glands are also there. These minor salivary glands may be the buccal salivary glands, may be the labial salivary glands, molar, palatine, etc. So function of the minor salivary gland is lubrication of the oral cavity. Also to initiate the digestion of the carbohydrates, because they contain the enzymes required for the initial digestion of the carbohydrates, and also to cleanse the teeth. So these are the different functions of the minor salivary glands. This diagram shows the anatomical position of the major salivary gland. You can see this is the parotid gland. This is located below and in front of the ear. Then the submandibular gland, which is present in the floor of the mouth, just close to the mandible. This gland is also called submaxillary gland, because some British anatomists also called mandible as submaxilla. So in relation to the submaxilla, the gland is also named as the submaxillary gland. Anterior to the submandibular gland is the sublingual gland, which is also present in the floor of the mouth. So what is the general structural plan of the glands? Salivary glands, they consist of the parenchyma, the stroma, and the duct of the glands. So here you can see the parenchyma of the glands and the stroma, which is connective tissue, and the ducts which are present in the substance of the gland, as well as larger ducts which are present in the connective tissue. So what about the parenchyma of the gland? Parenchyma is the compound tubulo-alveolar type of the gland surrounded by moderately dense connective tissue. This is the moderately dense connective tissue, and these are the lobes and the lobules of the glands, and these are the smaller septas which divide the substance of the glands into lobes and the lobules. So each gland is covered by the capsule, and this capsule sends in septas to divide the substance of the gland into the lobes and the lobules. So connective tissue composed of the capsule and the septas. You can see this is the capsulated gland. Remember, all the major salivary glands are capsulated, all the major, but there is no capsule around the minor salivary gland. So this may be important from your MCQ's point of view also. So all major salivary glands are covered by the capsule. This capsule sends in septas, and this septa divide the parenchyma into lobes and then lobules. This septa consists of moderately dense connective tissue. It also contains the larger ducts as well. So what about the microanatomy or histology of the glands? That the secretory unit consists of either the serous acini or the mucous acini, or it may be the mixed acini. So while you are looking into the microanatomy, you can see either serous or mucous or mixed acini. In addition to the acinus, you can also appreciate the myoepithelial cells, then the intercalated duct leading to striated, and then the excretory duct. So this is the diagram. You can see In this diagram, these are the mucus acini, and these are again the mucus acini having serous demi-lumen over here. So this type of acinus is called mixed acinus, and this is again the serous acinus. So in this diagram you can appreciate all the three types of the acini. This one is the mucus acinus, this one is the mixed acinus, and this one is the serous acinus. And this is the section of the serous acinus. These acini, their secretions are carried via intercalated duct into the striated duct. So this is the section of the intercalated duct. So what about the acinus? The acinus are this spherical structure consisting of secretory epithelial cells arranged around a central cavity which continues into the duct. So you can see these are the serous acini, having central spherical nucleus. Basal striations are also present. Myoepithelial cells are also present. They surround the acinus as well as the initial part of the duct also. So this is the intercellular secretory this one is the intercellular secretory unit, this, and then the myoepithelial cells. These are the mucus acini, and you can see the section of the mucus acini over here as well. And these are the striated duct, and these are the intercalated duct. So we now discuss all of these structures individually one by one. So first of all, the acinar cells, you know all that the acinar cells can be classified as the serous cells produce thin watery secretions, protein in nature. Mucus cells produce more viscous protein secretion, which is mucin in nature. And this is the section of the serous cell, the diagrammatic presentation of the serous cell. Serous cells stain darkly with both eosine and hematoxylin. So this is important point to remember. You can see this is the basally located rounded nucleus. Large number of the rough endoplasmic reticulum is present over here. In addition to this rough endoplasmic reticulum, there is well developed Golgi apparatus. Mitochondria are also present. So this RER along with the rounded nucleus give the basal portion of the serous cells a strong staining with the hematoxylin, that is strong basophilia. But while in the apical region you can see the zymogen granules are present. These are the apical granules which are called the zymogen granules. They stain with the eosine. So the apical region of the cell stain with the eosine, and basal region of the cell stain with the hematoxylin. So hematoxylin is because of the RER and nucleus, and eosine staining is because of these zymogen granules. So in addition to these features, you can also see that there is the basal infolding of the plasma membrane. In addition to basal infolding, lateral plications are also present between the cells, and this is the intercellular space, and these are the tight junction between the cells. So you can see tight junctions as well as the lateral plications and the basal infolding of the cells as well. You can also see other organelles are present in the perinuclear region, basally located nucleus. Electron microscopy shows basal infoldings and lateral folding that implicate with that of the other cells. Here you can appreciate this one as well. And intercellular canaliculus exists between the adjacent cells. So this is the intercellular canaliculus which is present between the adjacent cell. Also the basal folding. So this is the electron micrographic picture of the cell. You can see this is the myoepithelial cell, and this myoepithelial cell is present within the basal lamina of the secretory unit, cells of the secretory unit. And this is the serous cell and basally located nucleus of the serous cell, as well as zymogen granules are present. This is labeled as the actin filaments. These actin filaments are the filaments present in the cytoplasm of the myoepithelial cells, and myoepithelial cells having contractile ability helps to push the secretion of the cells into the duct, and also present in the initial part of the duct, where they also help to propel the secretion from the smaller ducts into the larger ducts. So here you can see these are the sections of the serous acini. In serous acini you can see strong basophilia of the nuclei which are present in basally located. Also you can see the dotted eosinophilia. These are actually the zymogen granules which stain with the eosin. This one is the striated duct. Next is the mucous cell. A mucous cell is basically a pyramidal shape cell. It is a larger cell which is stained very poorly with the eosin and hematoxylin. It stores the mucus in the form of the mucinogen granules. So you can see these are the mucinogen granules which are present, and these mucinogen granules pushes the nucleus towards the basal area, and this nucleus is flattened and it is present against the base of the cell. In addition to this, the large number of the RER are also present, and Golgi complex is also there. Intercellular application of the cell membrane, and also the tight junction between the cell also present over here. So this is the H and E staining of the mucous acini. You can see the large pyramidal shaped cells and having nearly empty appearance, and this empty appearance is because of the fact that usually by the process of the staining the mucus is washed from the cells, so cells appear empty and foamy, show foamy appearance. Also the basally located flattened nuclei are also present. These are the mucous acini. Next is the ductal system. You can see this area is the acinus, and here you can see different type of the serous acini are present, and myoepithelial cell over here is present. This is again another myoepithelial cell. This one is the initial part of the duct called the intercalated duct. Here the epithelium is a low cuboidal in nature. From here the mucus is pushed with the help of the myoepithelial cells into the next part, which is the striated duct. So in this striated duct, duct is also intralobar or intralobular in position, and here initially the epithelium is also simple cuboidal. Later on it becomes the columnar, and when the duct size of the duct increases, it can become the stratified cuboidal or stratified columnar, and the duct is here called the excretory duct. When these excretory duct reaches into the oral cavity and empty their secretion into oral cavity, their epithelium becomes the stratified squamous non-keratinized. In case of the parotid gland, the duct is called the Stenson's duct, and in case of the submandibular gland, the duct is called Wharton duct. So this is the ductal system: the acini, the intercalated duct, the striated duct, the excretory duct, which then lead via excretory duct into the oral cavity. Again you can see the different parts. This one is the mucus acini, and this is the section of the mucus this is the serous cell and section of the serous cell, basally located rounded nucleus and apical zymogen granules. Here the mucus cells are present having the flattened basally placed nuclei, and also you can see in this diagram that the mucinogen granules are present in apical region and basally located flattened nucleus. This is the section of the intercalated duct. This is a small cuboidal cell having a rounded nucleus, and this one is the section through the striated duct. The cells are over here are also the cuboidal or columnar in shape, but the important feature is that they have the basal infolding of the cell membrane, and in between these infoldings are placed the longitudinally oriented large number of the mitochondria, which gives in the light microscopy a striated appearance. That's why the duct is given the name the striated duct. In this diagram you can also see these are the acini, and then the acinus, and then the intercalated duct, then the intralobular smaller duct, then the intralobular larger duct, then the lobar duct, and then the main duct. So what we have written is that striated and intercalated ducts well developed in serous than in mucous glands. Striated duct, bicarbonate is taken in and chloride taken out from the lumen. They have a function in modifying the secretion. Intercalated duct, potassium into the lumen and sodium from the lumen producing hypotonic fluid, and the excreted duct do not modify the saliva. So this is important from MCQ point of view also. Structure of the striated duct, you can see these are the striated ducts. Here you can see these striations. These striations are because of the infolding of the basal plasma membrane and also the longitudinally oriented mitochondria, large number of the mitochondria which are present over here, and these two structures give eosinophilia and striation in the light microscopy. These all are the serous acini which are present over here. So the striated duct is intralobar in position. It is lined by the low columnar epithelium, also stained deeply with the eosin, and characteristic feature of the ion transporting cells because it is present in the cells where secretions are modified. So basal infolding of the plasma membrane is the reason for the striations, also the longitudinal orientation of the mitochondria between the enfoldings. So the duct system, that is the small intercalated duct leading to the striated duct, that leads to excretory and then to the main duct. So this is a beautiful diagram, H&E stain section of the serous, purely serous glands. These are all the serous acini having rounded, basally located nucleus and apical zymogen granules which are staining with the eosin. And this is the section of the intercalated duct. Here you can appreciate the rounded nucleus of the low cuboidal epithelial cells. All other these are the serous acini. So a little bit about the comparison. This is the serous cell, this is the mucus cell. Consistency of the secretion, thin watery. Here the thick viscous. Nature of secretory granules. Here zymogen granules are present. Here mucinogen droplets are present. Shape and position of the nucleus, rounded and central, flat and peripheral. Size of the lumen is small, size of lumen is larger. Appearance of the cell, boundaries indistinct in case of the serous and distinct boundaries in case of the mucus. Staining with H&E, it stains darkly both with eosin and hematoxylin, and it stains lightly. Function is the enzymatic function and its function is protection and lubrication. So examples are the parotid gland, and example of the mucus is sublingual gland. So this is the comparison. You know that this one is the parotid gland, this one is the sublingual gland, and this one is the submandibular gland. So you should also know another way of classification, that the parotid gland is purely serous in nature. Whenever you look into the slide, you can only appreciate the serous acini in the section of the parotid gland. In addition to serous acini, parotid gland also contain large number of the adipose tissue as well. And as you know from your knowledge of the gross anatomy, that the facial nerve is also passing through the substance of the parotid gland. So you can also see the section of the facial nerve as well if it is present in the parotid gland. Sublingual gland and submandibular gland both are mixed glands, but the difference is that submandibular is mixed but it is predominantly serous in nature, and sublingual gland is mixed but it is predominantly mucus in nature. So this is parotid, this is a sublingual gland, and this is the submandibular gland. Again, a beautiful section through your atlas. This one is the purely serous gland, having the serous acini, large number of serous acini which are present over here. And this is again a mixed gland having large number of serous acini as well as mucus acini with the serous demilunes over here. And this is again another section having the mixed gland Majority of the acinas are mucus in nature, but few of them having the serous demilume. These are the mixed acinas. So according to classification, mixed purely serous gland is the parotid. Mixed glands but predominantly serous in nature is mandibular. Mixed gland but predominantly mucus in nature is the sublingual gland. This is the section of the striated duct. This is the section of the at higher magnification, the mixed acinus. This is again a section at higher magnification of the mixed duct, and this is the large interlobular duct lined by the simple columnar epithelium. So must remember there are no intercalated or striated ducts in the substance of the sublingual gland. Again, you can see these are the serous acini. This is the mixed acinus having serous demilume over here, and these smaller flattened cells are that of the myoepithelial cell. This is the duct which is the striated duct. Again, you can see different sections over here. This is the serous acinus, and these are the serous acinus over here and mucus acinus over here, and this is a smaller duct which is the intercalated, and larger duct which is the striated duct, and the myoepithelial cells which are surrounding this ductal system. Here you can see the very clearly defining the myoepithelial cells which surround the acinus and initial part of the duct, and this is the section of the myoepithelial cells. You can see it's a flattened nucleus and large number of the actin filaments which are present over here. Again, another section through the serous gland. Here you can see large number of the serous acini, and this smaller one is the intercalated duct. It is lined by the simple cuboidal epithelium. Another beautiful diagram showing you the striated duct. This is the larger duct. So how you can differentiate between the intercalated duct and the striated duct? So you can see these all are the serous acini. Whenever there will be intercalated duct, it will have the diameter smaller than that of the acinus, and the striated ducts will be larger than that of the acini, and also lined by the cuboidal epithelium or the columnar epithelium. Another section taken to the excretory duct, and here the excretory duct is present in the septa, and this excretory duct is lined by the stratified columnar epithelium over here. Another section through the mixed gland. Here you can see the mucus acini and the serous acini. So in this section you cannot say whether it is sublingual or submandibular, because you can identify the slide while you are looking into the whole of the slide, not only to one portion. It may be sublingual, it may be submandibular, depending upon whether the mucus acini are predominant or the serous acini are predominant. Another section through the mixed acinus. Large number of the serous acini are present along with some mucus acini having serous demilume, and this is the striated duct present over here. So the gland is submandibular in nature. Again, another slide having large number of the mucus acini over here. So predominantly mucus gland is sublingual gland. This is the larger excretory duct. Again, a mixed gland having serous acini, mucus acini and mixed acinus, and the striated duct. Striated duct gives you a clue that it may be the submandibular gland. Another section through the mixed gland, the mucus acinus with serous demilume, the striated duct and the serous acini, and this section is also that of the striated duct, and here some blood vessel is present. Another beautiful diagram of the parotid gland having only the purely serous acini are present, and this one is the larger duct which is present in connective tissue septa. Another beautiful diagram, but it is a little bit confusing because a large number of the serous acini are present in it. It initially gives a look that it may be the parotid, but here you can see the mucus acini are also present. So mucus acini gives you a clue that this is a slide of the mixed gland, but predominantly serous in nature. So it is the submandibular gland. Again, another slide through the mixed, both the acini are present, the serous as well as the mucus, but you can see the large number of the serous acini are present. So again, this is the slide of the submandibular gland. Here you can see a beautiful striated duct lined by the stratified cuboidal epithelium, another section of the striated duct. This is another section of the striated duct. This is another section of the striated duct, and this smaller may be this one may be the intercalated duct. Another slide having both type of acini, the mucus and the serous, where proportion seems equal in this diagram, so you can't say whether it is sublingual or submandibular. Another diagram showing large number of the mucus acini along with smaller intralobular ducts, and this is the sublingual gland. So here I complete my today's class, dear students. These are the references: the basic histology by Laikus and Siddiq, the histology text and atlas by John Quirra, and histology text and atlas by H. Ross. Thank you very much for your attention for the today's class. In next class, we will continue the remaining part of the accessory glands associated with the digestive system. Till then, Allah Hafiz.